Osiejuk, and today I will be talking about doves. And if somebody told me 2 years ago that I would study doves, pigeons, I would, let's say, never in my life, not such simple birds. But here I am, and I will be talking about that because they are not so bad, in fact. Okay, so functions of song in birds are quite well recognized. And in birds in temperate regions where usually males are singing, they are usually sing to attract the mate or to defend territory. The situation is more complicated in areas such like tropics where many bird species are sedentary, and we have around 400 species which are producing songs in duets, so male and female, and even chorusing. And such function of such more complicated coordinated signaling are coordination of behavior or pair cohesion, so it's much more complicated. But to all this function, there are necessary some mechanisms which just allow, for example, to recognize if this is my species or not, to recognize individuals, to efficient territorial defense, for example. And today, in my talk, I will focus on these two mechanisms of species recognition and individual recognition in the species. And one more point about these stupid pigeons. So they are not learners, so they are much less flexible in sound production than such birds like ostriches or parrots or hummingbirds. So the question in background is how they are doing this, how they are territorial, how they attracting a mate without such flexibility. And the story started, in fact, when we were working together with our group in Cameroon some years ago on different species, different topics, but we were opportunistically recording all species around, mostly endemics, but also some common species like the kangaroo duck. And later, one of my master's students was using this data to find out if it's possible to discriminate between different individuals in the species. So we measured several things like the entire song's duration, number of notes. Also, we measured precisely the note duration, peak frequency, and pauses between notes. And it's possible to do this automatically for this species, so it's quite fast. And what we found was very interesting. We found that if we measured only the first 5 syllables, especially pauses between notes in the song, we were able to discriminate with 95% accuracy between individuals, and the data set was 43 individuals and over 500 songs, if I remember well. We also find out that if we measured sequences of 5 notes and along the song, so the number on the next axis, the 1 indicates notes from 1 to 5, 2 notes from 2 to 6, and so on. We found that the most information about the identity of individuals is only at the beginning of the song. So we can say that this initial part of the song is something like a fingerprints for this tambourine duck. So the aim of the next project, because of this initial study, was to find out if this is the same in all turtle wood dove species. We have 5 species in Africa. So they are found in sub-Saharan Africa, and in many places we have 2, 3, or even 4 species within the same area. They differ slightly in size, so these 2 on the left, so tambourine duck and blue-headed, are a little bit heavier than the other 3. And they differ also according to habitat selection, so the first 2 are prefer forest, especially the blue-headed one, the second of this picture. And the other 3 species like the blue-throated, the emerald dove, which is here also included, and black-billed wood dove. They prefer different types of woodland, savanna, miombo, and so on. Okay. And here we have songs of these old species. I will play only. I will play only the one because songs are quite long, so this will take 30% of my talk to play all of them. But as you can see, they are very simple. They consist of short syllables. Some species have some modulation in tones. Frequency is very low. So for this project, we were doing recordings in several countries in Africa. We were also catching birds using live tags to find out if we are recording the same individual, to check if they are changing something, either some during singing, because for most of the time, especially for this forest species, we cannot see the individual in the canopy. And we, of course, also finally did some playback experiments, and one of them is presented on the poster of Gosia. So what we find out? So if we look on the song duration and compare the species, we can see that they are different, but also overlap quite highly. So if we use the discriminant analysis to recognize only based on duration between the species, so it's not very good resonance. If we do this with the number of notes within the song, the discrimination analysis is a little bit better. And as you can see, most pairs of the species are not overlapping. If we do this with the frequency of maximal amplitude, again, the result of the analysis is a little bit above the 50%. But if we put all these three characteristics together, we can recognize, we can classify the sum to particular species with 82% correctness. So, based on this, we can say that they are, the sums are very similar between these five species, but there are some differences. The forest and bigger species have lower frequency songs, longer, with a higher number of notes. I'm not going to talk too much about amplitude now, but we also know that the song of the forest species is much better propagating, so you can hear as a human even from the distance like 500 meters. For the woodland species, it's twice shorter, but this will be important information for the later part of the talk. So maybe there's something more different, which is differentiating both species and the individuals if we look into details of the note sequences, similarly like we did for the tambourine dove. So if you can just look at the sonographs of all species, it's quite clear that the beginnings are much more different than the final parts. And the final parts usually, even if there is some change, it's quite constant. For example, it's faster and faster, but the duration of syllables are usually the same. And in the initial part, usually we have groups of syllables, some kind of blocking, or in the such species like emerald wood dove, we have also some slow modulation of frequency. This graph are showing this in detail, so you have the colors which all the time indicates from the deep green to the yellow, from forest to the savanna habitat. So note duration is always decreasing for all the species from the beginning of the song. The same is for most of the species with the pulse duration except one. If we look at the peak frequency, usually it's also decreasing within the song, but one of the species, namely the blue-spotted one, the Tortuga has a tricky snake shape on this graph. And what was really the most interesting for us is the relative amplitude. So on this graph you have the zero on y-axis means that this is the loudest part of the song. So each syllable, each note was just measured and compared to this part. So as you can see, the beginning of song of most of the species, and especially these living in savanna, is really quiet. Here we have just smoothed the line which averages for the 500 songs, but the maximum lowest value we had was minus 32 decibels at the beginning. So this means that the range, the active range of the signal for the whole songs and for some part of the songs are really different. And of course, this was significantly different between the species if you compare initial and final part of the song. So let's move to more discriminant analysis. So on this slide, you can compare efficiency of discrimination for all species, the darker part, and for discrimination of different individuals within the species, based on these three basic song characteristics, so only the number of notes, duration, and maximum amplitude. So it's pretty nice. If we do the same, but we consider only the pauses between the following initial and final syllables, and we stay with comparison of different species and discrimination to different individuals within the species, we can see quite a nice pattern. Namely, in the forest species, we have a situation where is the more identical information at the beginning of the song, and in these species which are singing with lower amplitude, and the last part of the song is more relatively more louder, the final part of the song contain more signature about ID. And here is also this comparison of this initial and final part with the relative amplitudes. So to summarize these results to conclusion. So the four species have lower frequency songs, longer songs, only slightly lower amplitude at the very beginning, but usually this is only the first 1, 2, or 3 syllables, not the rest. The savanna species has higher frequency song and shorter, and the beginning of the song is really substantially more quiet than the middle and final part of the songs. So it seems that there's difference between these two groups of species with locating identity information within the song. So it seems that the evolution of song in this distinct group of doves was shaped by the body size, especially with the frequency, but also with the habitat where they live. And it seems that there is a difference in differentiation in these songs. They seems to maximize the information transfer about the species ID, let's say, which is useful for discriminating between one and the other species, especially if you are living in the same area. And if information about the identity seems to be optimized. It's not at the maximum level as it could be if these parts which contain this information would be louder. So this was the basic study which was used for us to prepare experiments. So we are after the experiments with 3 species in which we tested how they are responding to their own species and to the other species. In SYNCPA3, AlloPA3, we also use some different mixed songs which take part from one species and from the other. So some of the results are from the poster and some are just, we finished preparing them a few weeks ago. And in this next year, we are planning to do the experiments with neighbors, just classic neighbor-stranger discrimination, but we will be also modifying some parts of the song to find out how birds are responding if we change the part which we suggest is used, for example, for coding ID. And at the end, I would like to thank institutions which are supporting us in our study.